Welcome to our COVID-19 in VR series. Today we have Dr. Mike Kuiper, actually joining us from Australia. Yeah, thanks so much for joining us. And yeah, if you want to share a little bit about your work on the coronavirus, which we first saw with you
Absolutely.
about a year ago, right?
That's right. Yes. Thanks, Steve. It's great to catch up again. And it's great to see that we can talk across the Pacific, mind you. It's quite the distance. And it just feels like I'm right there with you guys. Of course, a lot has happened, but I guess first a bit of an introduction about myself. Uh, my name's Michael Kuiper. I work as a biomolecular modeler at CSIRO, which is an Australian research organisation. And we basically do all sorts of areas of science. I do biomolecular modeling. I help researchers who are actually working on experimental results looking at vaccines and vaccine development. In particular, over the last year, we've been focusing a lot on monitoring the evolution of the SARS-CoV, particularly looking at mutations and how they may affect the virus. So I thought
Yeah.
I'd share today a little bit some of the work that's been going on in the last year, and in particular, recent developments. As many of you are no doubt aware, there's been some concern over a UK variant and also a South African variant. So we've been looking at these closely and trying to determine just how important these may be in terms of the vaccine.
A year ago we had you on the live stream. So if anybody
Mm.
wanted to check it out, they could do that. But this is a very similar structure with a few differences, and it's there's a lot more than just that one, but it's all these key differences we're going to be talking about how the virus changed between a year ago when we first got the original samples of what it looked like to where we are today with so many people around the world getting it. It's just mutating
Mm.
everywhere. But yeah, what does that actually mean? And you've got this nice table here, right? Yeah, I see some familiar names like the D614G mutation.
Ah, yes. Yeah. Yeah, there's so many, I guess, that are happening now as the virus has been infecting so many people, as people themselves are developing immunity, as people are being have a vaccine. That's great because they have some immunity, but it does add some selection pressure to the virus. So this is why it is mutating, and it's not necessarily always going to be a bad thing. The virus can also mutate to become a less virulent form as well. So that may be something that could occur down the track as well. However, we must be vigilant, so we must keep looking at this thing and keep an eye on this. What I have here today is a model that you've no doubt seen many times before. It's an important part of the spike protein. In the blue, we have the receptor binding domain, which binds to the human receptor ACE2. Now, the reason why I have a smaller model here is because when we actually do a simulation, a molecular dynamics simulation, it's much easier to do just a small section of the virus and the key interaction rather than the whole thing. We have done the whole thing, the whole spike protein, but of course it's much slower. So I just have a few frames here from the simulation, so you can sort of see that I can just click through. And this just gives us an idea, when we do this, of where the residues actually like to be. So for instance, here at the start of the simulation, I've just highlighted one of the residues here, which is lysine 484, which we see in the South African strain, where we go from a glutamic acid to a lysine. And at the moment, it doesn't look like it's doing much. It's floating out here somewhere. But during the course of the simulation, we actually see the ACE2 come to actually bind to the receptor binding domain. And all of a sudden, this mutation starts to make a lot more sense in that it's creating now a Saltbridge between lysine 484 of the receptor binding domain with glutamic acid 75 of the ACE2. So these are the sorts of things that we're looking at. What are the favorable interactions which are occurring, which may strengthen the interaction and the specificity of ACE2 with the receptor binding domain. But I did also want to bring up some of the other work that's going on around the world. So, of course, it's just not all visualization and modeling, but a lot of people do experimental work based on this. So the amount of work on COVID has just been enormous. There's literally something like 2,000 papers published a week. So it is quite tough to keep on top of all that literature as well. But a few papers do come to mind, and I wanted to bring this particular paper, which was a tour de force. This is deep mutational scanning of SARS-CoV-2 receptor binding domain. Reveals constraints on folding and ACE2 binding. So basically what they have done in this particular paper is they recombinantly expressed different versions of the receptor binding domain and then measured the affinity to the ACE2 protein. And what they actually did was they looked at each amino acid which potentially binds with the ACE2 domain and mutate that for every other amino acid possible. So that's an incredible amount of work, and I think it just goes to show what we can do as a scientific community when we put our heads together. So this work has been incredibly useful.
Very
So
much
if we just look
an emphasis on the deep mutational scan. They took every residue and then mutated it for every other residue to see how they would all bind. That's amazing.
It is, and this basically is a tabulated result of that. So I just wanted to bring your attention to this figure here, which basically summarizes a lot of that work. So each little square that you see here is representative of an experiment, of a actual recombinant protein and an actual couple of experiments there. Wherever you see blue, that's where, or more bluer colors, that's where a mutation has occurred in a tighter binding. Wherever there's red, it's gone the other way, where it's actually a detrimental binding. So when we actually start to look at some of these down here, in particular, let me highlight this position. If we can see here, position 501, if we look at that there's a bit of a blue patch here. And if we look across, that matches to phenylalanine and tyrosine there. And if we look at our table, of course, over here, of our new mutations, this is, of course, the N501Y. So these mutations that we're seeing in the UK and the South African variant were actually predicted from these earlier experiments, which I think is really quite remarkable and gives us a great idea of where potentially this can mutate and which direction it could go to, what things to look out for.
Wow. So I think it already predicted. So this one was a pretty strong blue indicator, and we've already seen that in both South Africa and UK. Is that independently? They both emerged from their own strains that diverged a long time ago, and they independently did that?
I'm not sure. I'm not sure about that one. Okay, because that'd be really
interesting. Yeah, if they were sort of convergently mutating on some of these.
It could be. It could be very much that sort of scenario. But I think this just goes to show how together we can bring all this information together from multiple sources to try and truly understand what's going on here. When you think about how many, if you like, experiments are going on in the world, in that how many people are actually infected. A year ago, we had, just tens of thousands of people infected. Now we have literally tens of millions. So that automatically increases the probability for more selection, more mutations to occur.
And we have a giant petri dish right now.
We really are. So you can see another one down here, 453. And that is, if you look across, that is F, which is very similar to Y. Actually, it is F. Okay, so F is a stronger binder, And do you remember what 453 is? Why
What?
453F? That's
That wasn't one of
that's
the
the
mink,
mink
mink ones. Yeah, that is the
that's
mink.
the
Okay.
mink mutation. Yeah.
Yeah,
So
there
again,
we
you
go.
can see in this experiment they also predicted the mink mutation.
Wow.
So that's actually quite interesting to see. And of course, I saw you did an earlier piece on the mink mutations, and that makes sense with the ACE2 because where you have the histidine binding at that interface, you actually have a tyrosine there instead, and that makes a nice pi stack as well. So yeah, I think this is really fascinating for us to be looking around at the high quality research that is going on out there and paying attention to it, because I think there's a lot of insights that people are already finding, but it's because there's so much information out there, it's really hard to bring it all together sometimes.
Definitely, yeah. If we could, just bring everybody into VR and throw up our papers and our data and get together, understand what's going on. It's what we want to see in the world.
Yeah, and I think that's what I love about the VR, because when you're sharing that experience with someone, it's no longer just a little blip on a graph. It's you can actually point at it and and highlight that residue and highlight, for instance, these two residues here, which seems like an important interaction.
The Brazilian mutations as well. Uh, have you been looking into those? I haven't really looked at each one in detail.
Ah, yeah, there was one that they found in Japan, right? That
Hmm.
originated in Brazil. And I think one of those was K417T, I believe. So 417, of course, occurs down here. Now, what I thought about that was, I'm not sure why that particular mutation, and in the South African variant, it mutates to asparagine. My gut feeling is that that's actually may be a detrimental for the binding. So I wouldn't have
Yeah.
expected that.
Well, it looks like it's a pretty light color, just like light.
So yeah, when I actually first looked at this, I was misreading that column and I thought it was one of these darker red ones, which would. But having said that, and the reason I thought that lysine may be preferred over either threonine or asparagine was that potentially you could have this Saltbridge interaction here. So you can see this nice little sort of interaction there, which over the course of dynamics, you can see occasionally gets pretty close.
Yeah, interesting.
So I would have expected this mutation to stay a lysine myself because of the let me just label that. So that's aspartic acid 30 there. So I was a bit surprised to see that mutation arise, but of course it's a combination of everything, and it may be a much more subtle interaction. There may be solvation effects as well. So one of the things you don't see in this model, because we take it out, are the water molecules, of course. So the water molecules are often forgotten, I guess, but they have a very important role for interacting between charges, interacting between the interface. And sometimes you will see bound water molecules as well, which are sort of important for the overall interaction. Of course,
Sorry, no, keep going.
Oh, no, go ahead.
Well, I was gonna ask, like, in these like benign or let's just say neutral, lighter colored mutations that might not change the interaction too much, or they may. How much does this sort of evade the immune system, right? If there's enough, sort of arbitrary changes where it just goes from like white region to white region, like, is that going to make it harder for antibodies that recognize the previous iteration?
That's correct. And that's the other half of the equation, of course, is it's not all about the ACE2 interaction. It's about a combination of evading the immunity that it's out there. So maybe that's that's really what it's doing. It's helping evade the immunity without affecting the interaction too much. So you're absolutely right there. So hard to say at the moment what it is. So I think that's where people will start to look at these mutations, do more experiments with the antibodies, and trying to understand just how much they can evade that particular antibody or therapeutic, monoclonal, whatever the case may be.
Mhm.
Yeah.
Do you compare things to like Bamlanivimab? Sorry, I probably butchered the name, but I'm talking about the Eli Lilly antibody drug, Bamlanivimab.
uh, we've started looking at a way to, I guess, try and filter this by looking at the structural information when you actually look at an antibody receptor interaction. You can usually see how the antibody binds to that particular receptor, and then you can sort of just highlight those residues, and then you will know that if those residues change at the interface, then that's probably likely to affect the kinetics of the antibody binding. So you'd have to look at the experimental results, because you really need that crystal structure, I believe, to get a good
Mhm.
understanding of how that interacts. And actually, that's what I've been using a lot of VR for now these days, is reading in one of those structures and then basically pulling it apart and just seeing how the antibody is sticking to the particular protein, usually the receptor
Oh,
binding domain.
nice. It's awesome.
Yeah. This is what I like about molecular dynamics simulations as well. When you look at a crystal structure, you see it as a static image, as a statue, if you like. And of course, in the real world, things move around and things are very dynamic. Unfortunately, the amount of computer simulation that we can do is limited. So, for instance, if I wanted to simulate a few nanoseconds of molecular dynamics, that'll probably take me a few hours to do. We can typically, on a system this size, we can easily do 100 nanoseconds a day. But if you think a nanosecond is a billionth of a second, so if I'm trying to replay the trajectory, if I'm seeing 10 nanoseconds a second back in the playback in the trajectory, it's still going to take me 3.1 years to sit and watch the whole trajectory for a second. So that just gives you an idea of how much is going on in a second at that atomic scale. A lot is moving, a lot is wiggling and jiggling. So it's really quite remarkable how much movement goes on in a second.
Definitely.
Yeah. But yeah, that's what I really like about these molecular dynamic simulations, is they give you a chance to see how the protein structure can settle and how the residues will move around and interact with one another. Things that didn't look too close to begin with. So, for instance, if I come back to our original example here at the start of the trajectory, if I move that back, you can see that I'll just measure the distance here. So you can see at the beginning of the trajectory, it was something like 10 angstroms. So you wouldn't expect it to be particularly close. But, a second later on the next frame, we're actually 20 angstroms. So you can see there's a lot of movement. And that's because when in the start of the simulation, we don't have any constraints. We just allow these to move and they will naturally fluctuate. So then when I go to the next frame, you can see they're closer again. And then here they're actually making a hydrogen bond contact. So that's, I think, what's so powerful with molecular dynamics is you can actually get an understanding of what the residence time of particular interactions are, whether an interaction is strong or whether an interaction is weak, and what the effects of some of these mutations may actually have. So that's sort of where we look at with this.
Yeah, it's awesome to see them really like coming together because like you said, when you look at the crystal structure, it's like you only
Uh-huh.
get one picture, one snapshot of what's going on. You don't get to see the full story. And even with sort of longer simulations, we're never going to see the true full story, but we could at least keep getting closer and closer by doing these simulations and seeing how things move. What are the states that it likes being in? Does it have any conformational changes? Going
Mm.
on there. So yeah, it's great to see
Ab-
dynamics.
Absolutely. And that's the caveat with molecular dynamics. It really is just a snapshot into what we think is going on. And it's a very small snapshot. And perhaps it gives us some insight into what's going on. Typically, what this allows us to do is highlight some potential interactions, which we can then further explore with experimental results. And then the experimental results will often either confirm or deny a particular hypothesis, or at least suggest that it could be true. And then we can move on from there to the next stage of the modeling. Maybe we improve the quality of the model. Maybe we can suggest some other experiments to do to really get to the bottom of what's going on in a particular interaction. Very much like this cell deep mutational paper. That is really a tour de force of experimental work, and that can feed directly into our understanding of this sort of these sort of interactions, which are crucial to the COVID pandemic.
Right.
Cool. Yeah, we'd also like to thank Tyler Starr and Jesse Bloom for contributing the awesome work with the deep mutational screening of SARS-CoV-2 receptor binding domain. Crazy work what they've done to show all the things that could happen in terms of mutations. Even stranger that we're seeing this in the real world happening today, and we're able to keep real time on top of it by going into virtual reality from around the world. Thank you, Mike, for joining us all the way from Australia. It's been lovely as always to see, your structures, your simulations, your thoughts and your insights. Thanks so much for, hopping into VR and using Nanome.
Thank you, Steve. It's always a pleasure to catch up in VR, and it's so effortlessly to be able to do that these days, and especially over the distance. I can't believe that this connection works so well, and we can have the opportunity to discuss the latest findings.
Yeah, thanks everybody for watching our COVID-19 and VR series.
Thanks.